Welcome students, here is Jain with a new topic of cell biology that is called as the cell wall. The cell wall is the feature of the plant cell. This is the main differentiating feature between the plant cell and the animal cell, that in the plant cell wall is present, while in the animal cell wall is absent. In the plants, the cell wall is made up of cellulose, hemicellulose and pectin, and also there are few substances like the lignin, suberin, cutin, which are also deposited on the cell wall. If we look about the bacteria, then bacteria are also included in the plant kingdom only. Why? Because they are having the cell wall, but now they are kept in a separate phylum known as the Monera. But still the bacteria is having the cell wall, and the cell wall is made up of peptidoglycan. Peptidoglycan means it is made up of proteins, polysaccharides, etc. If you look on the fungi, then fungi are also having the cell wall, and the cell wall of the fungi is made of a substance known as the chitin. Now you can see here that how do the growth of the cell wall occur. The growth of the cell wall occur by the help of the two methods. One is known as intussusception and the another one is known as apposition. If the cell wall material is deposited in the form of the granules, then this method is called as intussusception. And if the cell wall material is deposited in the form of the layers, then it is called as the apposition. Now the primary cell wall is formed by the intussusception method, while the secondary cell wall is formed by both methods, that is both by the intussusception as well as the apposition method. Now you have seen that the cell wall is made up of the cellulose, hemicellulose and the pectin. Now we must know that where these substances are deposited, where these substances are formed, cellulose, hemicellulose and pectin are synthesized in the Golgi body, and these substances form the cell wall. Now if we see the structure of the cell wall, then the structure of the cell wall is composed of the four components, that is the middle lamella, primary cell wall, secondary cell wall and tertiary cell wall. But this tertiary cell wall is rare and it is found at few places only, right? So the major components of the cell wall are the middle lamella, primary cell wall and secondary cell wall. I have drawn a diagram here. You can see in the diagram also that here at the center of the two cells, suppose this is the cell A and this is the cell B. These two cells are joined together with a cementing layer, and this cementing layer is known as middle lamella. First see the position and then we'll be discussing in the detail also. This is the middle lamella which is present in between the two cells. This is cell A, this is cell B. In between the two cells, what is present? Middle lamella. And on either side of the middle lamella, this is the middle lamella. On either side of the middle lamella, this and this is the primary cell wall. This is also the primary cell wall. So the location of the primary cell wall is that it is present on either side of the middle lamella. And inner to the primary cell wall is present the secondary cell wall, here also and here also. A secondary cell wall is itself composed of three layers. One is known as S1, S2 and S3. So let's discuss the structure of the cell wall in detail. The structure of the cell wall consists of four parts: middle lamella, primary cell wall, secondary cell wall and tertiary cell wall. Number one, middle lamella. Now middle lamella is a cementing layer, present between two cells. This is the cementing layer which is present between the two cells. It is made of calcium and magnesium pectate. It is made up of calcium and magnesium pectate. Middle lamella gives hardness to raw fruits. Middle lamella gives hardness to raw fruits. If you have seen a unripe mango, you can see that it is very much hard. It is because it has the middle lamella. But as soon as what happens, the fruit ripe, the middle lamella dissolve. In ripened fruits, middle lamella dissolve, and so they become soft. Okay, so this was the first component, middle lamella, which is present between the two cells. It is made up of calcium and magnesium pectate, and the unripe fruits are having the middle lamella. That's why the unripe fruits are hard, while in the ripened fruits, middle lamella dissolve and they become soft. The second component is the primary cell wall. Now see the location in the diagram that it is found on either side of the middle lamella. I will write here, found on either side of middle lamella. It is thin and elastic, and it is made of cellulose, hemicellulose and pectin. Okay, so this was about the primary cell wall, which is the second component of the cell wall. Now coming to the third component, which you can see that it is present on either inner side of the primary cell wall, and it is called as secondary cell wall. It is thick and non-elastic and present on inner side of primary cell wall. It is made of cellulose, hemicellulose and lignin. You can mark here that here lignin is deposited, and that's why the secondary cell wall become thick and non-elastic. But one thing to be noted here is that meristematic cells are devoid of secondary cell wall. I have written in short secondary cell wall. Meristematic cells, the dividing cells, are devoid of any secondary cell wall. And the third one, which is very much rare, that is the fourth one, tertiary cell wall. It is not commonly found, it is rare and found in tracheids of gymnosperm, and it is made of cellulose and ghylin. Carbohydrate is deposited on it. It is made of cellulose and ghylin. The ghylin carbohydrate is deposited on it. So altogether we can say that the cell wall is composed of four components: the middle lamella, the primary cell wall, secondary cell wall and the rarely found tertiary cell wall. Okay, now let's have a look on the second one, that is the ultrastructure of cell wall. In ultrastructure, we will explain that the cell wall is composed of the cellulose. Have a look here, ultrastructure of the cell wall. If we look the ultrastructure of the cell wall, it is composed of two things, fibrils, and the second one is known as the matrix. Matrix is the ground substance in which the fibrils are present. A network of the fibrils are present in the ground substance known as the matrix. Now what happens? There are many macrofibrils. There are many macrofibrils in the cell wall. Out of that many macrofibril, I will be talking about one macrofibril. So remember, one macrofibril is equal to 250 microfibril. Now, one microfibril is made of 20 micelle. One micelle is made of 100 chains of cellulose, and one chain of cellulose is made of at least 3000 to 6000 glucose units joined together by beta 1,4 linkage. This is the ultrastructure of the cell wall. The structure of the cell wall never suggests that where is the cellulose present, but the ultrastructure of the cell wall is seeming clearly that where is cellulose present, and it also suggests that cell wall is made up of cellulose. Again, the ultrastructure of the cell wall is made up of fibrils and the matrix. There are many macrofibrils in the cell wall, but out of that many macrofibrils, one macrofibril is equal to 250 microfibrils, one microfibril is equal to 20 micelle, one micelle is equal to 100 chains of the cellulose, and one chain of the cellulose is made up of at least 3000 to 6000 of glucose units joined together by the beta 4 linkage. By the ultrastructure of the cell wall, you can see that how complex is the structure of the cell wall. Now, we know that the lignin, is deposited on the cell wall, right? But the deposition of the lignin, here I am writing deposition of lignin is not uniform on cell wall, and there are few places left on cell wall where lignin is not deposited. These places are called, very important, called pits. Are you getting? We know that on the cell wall lignin is deposited, but I am saying that the lignin deposition on the cell wall is not uniform. There are at least few places found on the cell wall where the lignin is not deposited. These places are called as pits. Say for, if this is the cell wall, at whole places the lignin is deposited, but there are few places, say for this one, where the lignin is not deposited, and these places are called as what? The pits. Now, pits are of two types. Very important. Pits are of two types. One is known as simple pit, and another is known as bordered pits. Now, what is the chief difference between a simple pit and a bordered pit? These simple pits are having uniform diameter, throughout the length. I will make the diagram also in few couple of minutes, then it will be able to be explained. Are having uniform diameter throughout length. But if you see the bordered pits, they are not uniform in diameter. The diameter increase from inside to outside. Are you getting? What is the chief difference between the simple pit and the bordered pits? That they are having the same diameter throughout the length, and they are not having the same diameter throughout the length. The diameter increase from inside to outside. Now have a look on the details of these pits. First of all, you must know that pits are always present in pairs. Okay, pits are present in pairs. This point to be remembered is that pits are always present in pairs. Say for this is one cell and this is another cell. So the pit of this cell is in front of the pit of the another cell. So always the pits are present in pairs, and the pits of two cells are separated by pit membrane. The pits of the two cells are separated by pit membrane, and this pit membrane is made up of middle lamella. This pit membrane is made up of middle lamella. Say for if we talk about a simple pit, just now I have said that a simple pit is having uniform diameter throughout the length. Say for if this is a pit, simple pit, this is pit of one cell, this is pit of another cell. You can see that they are having the same diameter throughout the length, and they are separated by what? A pit membrane. So the diameter here also is same, here also is same, here also is same, here also is same. You can see that the diameter is same. But bordered pits are different. The diameter of the bordered pit is not uniform throughout the length. Say for this is one pit like this type, it is having diameter like this type, and this is another pit. You can see that it is seeming to be just like a inverted funnel. You can also draw this diagram like this type. This is one pit, this is another pit, and in between these two pits is present what? A pit membrane. This is what? Pit membrane. And what happens in the bordered pit, that this pit membrane swells from the center. In bordered pits, this pit membrane swells from the center, and this is called as torus. This is called as torus. Torus is acting as a valve. Torus is acting as a valve. Why? Because through the pits, the substances are exchanged between the two cells. So these substances are actually moving from one cell to another cell. So the control on this transport of the substances is done with the help of the torus. That's why we can say that the torus is acting as a valve. Torus is acting as a valve. So this is the bordered pit and this is the simple pit. And one most important thing must be noticed here, that inside pits, what are present? Plasmodesmatas are present. Plasmodesmata, very very important. Plasmodesmata present, and due to these plasmodesmatas which are present in the pits, the two cells are in contact with each another. The two cells exchange their cytoplasm just because of these plasmodesmatas. So we must know that inside the pits what are present? Plasmodesmatas are present, and through these plasmodesmatas the exchange of the cytoplasm occur between the two cells. So this was about the simple pit and the bordered pits. One more thing, when we are talking about the cell wall, we must know that on the cell wall certain substances are deposited, and we must know about the nature of these substances. Say for suberin, we know that suberin is also deposited. Now suberin is a mixture of fatty acids. It is a mixture of fatty acid and it is deposited on the cell wall. Likewise, You must also know about the cutin. Cutin is a wax-like fatty substance, and it is also deposited on the cell wall. The third one, that is also lignin. You must also know about that lignin is what? So nature-wise, lignin is polysaccharide. Lignin is polysaccharide. So these substances can be deposited on the cell wall. These substances can be deposited on the cell wall. And at the last, we must also know about the function of cell wall. We must know about the function of cell wall. That what is the chief function of the cell wall? The chief function of the cell wall is protection. Because it is present outside, its main role is protection, and it is also providing the shape. It is also providing the shape to the cell wall. One thing at the last, that is, we must know that what is cytogenous cavity, how this cavity is formed, and we must also know about the lysogenous cavity. Many times this question is asked in the medical entrance examination, that what is cytogenous cavity. You must know that cytogenous cavity is formed by separation of cell wall. Cyto means what? Separation, splitting, separation, splitting. So it is formed by the separation of the cell wall. The best example of the cytogenous cavity is the resin canal of pinus. And the lysogenous cavity is, lyso means what? Lysis means it is formed by breakdown of cell wall. And the best example of the lysogenous cavity is the oil glands of citrus fruits. You know what is citrus fruits? Say for the orange or the lemon, these are the citrus fruits. So the oil glands are present in these fruits. Oil glands of the citrus fruits have lysogenous cavity, and the resin canal of the pinus is having the cytogenous cavity. So dear students, this was the explanation of the cell wall. If you have liked this video, subscribe my channel. Also like this video and share this video. And if you are facing any doubts regarding the biology, then you can comment in the comment section. I will be liking to answer to your questions as soon as possible. Thanks a lot. We'll be coming back with new topics. Goodbye.